I will be discussing eukaryotic gene expression, and I will give an overview of this eukaryotic gene expression in this lecture. Uh, Gene expression in case of prokaryotes has very classical examples of lac-operon and TRP-operon. We will see what are the differences in eukaryotic gene expression. Regarding prokaryotic gene expression, I have delivered that lecture earlier. And later on, in another lecture, we will see heterologous gene expression. So basically, this expression, when we say something about expression, it means transcription and translation. But the main control we will be discussing during this lecture is at the transcriptional level, because although there is some sort of control at the translational level, but the major control still they are at the transcription level. Transcriptional ground state uh, can be defined as the inherent activity of the promoters and transcriptional machinery in vivo in the absence of any regulatory sequences. And this transcriptional ground state is different in prokaryotes and in eukaryotes. In case of bacteria, RNA polymerase has access to every promoter and can bind and initiate transcription at some level of efficiency. In the absence of activators or repressors. Although activators or repressors, we see some examples, but transcriptional ground state is generally non-restrictive. Whereas in case of eukaryotes, the promoters are generally inactive in vivo in the absence of regulatory proteins. Means they require certain sort of activation, and transcriptional ground state is restrictive. And this is quite obvious from the chromatin structure in case of eukaryotes, in which we see the DNA lies hidden in the different types of proteins, and nucleosomes are there, and then other structures, hierarchical structures, they are present in case of eukaryotic DNA. So this fundamental difference gives rise to at least four important features that distinguish the regulation of gene expression in eukaryotes from that of prokaryotes. The first one is that the eukaryotic promoters, access to these promoters is restricted by structure of chromatin. And activation of transcription is associated with many changes in chromatin structure in the transcribed region. Second difference is that although eukaryotic cells have both positive and negative regulatory mechanisms, positive mechanisms predominate in all systems. Thus, given that the transcriptional ground state is restrictive, virtually every eukaryotic gene requires activation to be transcribed, while this is not the case in case of prokaryotic cells. Third point is eukaryotic cells have longer, more complex multimeric regulatory proteins than do bacteria have. And the last one is that transcription in eukaryotic nucleus is separated from translation in cytoplasm in both space and time. And you know that there is a nucleus present in the eukaryotes where transcription takes place and translation takes place in the cytoplasm, whereas no such boundary exists in case of prokaryotes. And even uh, the transcription is going on, the translation begins in case of prokaryotes. Transcriptionally active chromatin is structurally distinct from inactive chromatin. About 10% of the chromatin in typical eukaryotic cells is in more condensed form. This is called as heterochromatin, and this is transcriptionally inactive. The transcriptionally active chromatin is less condensed, and it is called as euchromatin. And these transcriptionally active chromosomal regions, they can be easily detected by DNase 1 because it tends to cleave the DNA which does not have much proteins bound, it means there are more chances of uh, transcription activation. So transcriptionally active chromosomal regions have increased sensitivity to DNase 1 nucleases. Many hypersensitive sites correspond to binding sites for known regulatory proteins, which is quite obvious that the hypersensitive site means they are uh, free to bind to nuclease in this reaction. So it means that that particular part of the DNA is present in the open form where some sort of proteins can bind. And relative absence of nucleosomes in these regions may allow the binding of these proteins. Nucleosomes are entirely absent in some regions that are very active in transcription. These ribosomal RNA genes, uh, they are transcribed uh, much more than any other gene. So their major part of the transcription goes on in transcription of the ribosomal RNA genes to make ribosomal RNA, and it has been found that nucleosomes are absent in this part of the DNA which contains ribosomal RNA genes. And transcriptionally active chromatin tend to be deficient in histone H1, which binds to the linker DNA. There are four types of histone molecules: H2A, H2B, H3, and H4. These two molecules of each, they make this nucleosome. And the H1, it is attached to the uh, knot of the DNA, where all these four types of DNA are within the wound around DNA. So, in order to make it convenient, make the transcription convenient, these transcriptionally active chromatins, they are deficient in H1. Histones within transcriptionally active chromatin and heterochromatin also differ in their patterns of covalent modifications. The core histones of nucleosome particles are modified by methylation of lysine residues, phosphorylation of serine or threonine residues, acetylation or attachment of ubiquitin, etc. 5-methyl cytosine of cytosine residues of CpG sequence is common in the eukaryotic DNA. But in transcriptionally active chromatin, that part tends to be undermethylated. So CpG sites in particular genes are often more undermethylated in cells from tissues where genes are expressed than those where genes are not expressed. Chromatin is remodeled by acetylation and nucleosomal displacements. Acetylation and deacetylation of histones is one of the major component of this uh, chromatin activation for transcription. Amino terminal domains of the histones are generally rich in lysine residues, and these are acetylated by histone acetyltransferases, HATs. There are two types. One are the cytosolic type of HATs. These histone acetyltransferases, they acetylate newly synthesized histones before the histones are imported in the nucleus. And subsequent assembly of histones into the chromatin is facilitated by additional proteins such as CAF1 and NAP1. The nucleosomal histones are further acetylated by nuclear histone acetyltransferases. The acetylation of multiple lysine residues in amino terminal domains of histones H3 and H4 can reduce the affinity of entire nucleosome for the DNA, thus allowing the transcriptional machinery to bind. Deacetylation of histones takes place with the help of histone deacetylases, where transcription is not required. This is a figure of showing the histones, nucleosomes. These inside the yellow ones are the 4 histones, which are H2A, H2B, H3 and H4. They are small molecular weight proteins, and these are the number of amino acids, and you can see they are rich in lysine and arginine residues. And this H1 is present here at the knot. So this is the hierarchy of nucleosome, and this is the modification of histones. There are different histone modifications such as methylation, phosphorylation. These two are showing here, and these are different types of histone modifications that exist. Few of these examples are here. There are more examples in nature. And these enzyme complexes actually catalyze chromatin structural changes associated with transcription. So these are the ones which are involved in histone modification. And uh, these are the sources like yeast and other eukaryotes, and these are their activities. Their names, there is no such classical classification of these names. They are given arbitrarily. Then there are histone movement replacement enzymes that require ATP. So there are different protein families like IS, W1 family, SW1, SNF, and so on, and they help in nucleosome remodeling, transcriptional activation. And the third one is the histone chaperones that do not require ATP, and they also help in deposition of certain types of modified histones uh, during the transcription. So, general transcription factors are first involved in transcription of RNA polymerase II in case of eukaryotes. This Tata box, this sequence is a typical of the eukaryotic promoters at -30, and this transcription is initiated at this +1. This is the sequence, and various regulatory sequences may be present upstream or some even downstream. But the main thing is that this Tata box is bound by a Tata binding protein, and this Tata binding protein then allows the binding of other transcription factors, which are referred as transcription factor 2B, and similarly 2F, 2E, 2H. So all these bind, they come in place in a certain sequential manner. And finally, this polymerase 2, along with transcription factor 2F, it comes in the picture, and then 2E and 2H. And when this transcription has to be initiated, uh, the C to C terminal domain CTD of the RNA polymerase 2 is phosphorylated with the help of transcription factor 2H. So transcription factor 2H has important function of in initiation of transcription of the RNA polymerase 2 in eukaryotic cells. But once this is phosphorylated and transcription is initiated, remaining factors, they remain there, and the transcription factors 2H and 2E, they are also released, and this RNA polymerase along with the transcription factor 2F and only a few factors, they continue with this elongation of the transcription, and finally termination of transcription takes place at the 3' polyadenylation, with the 3' polyadenylation, cleavage and polyadenylation step that takes place. So this is the hierarchy of, or the sequence of, the general transcription factors. But we will see what are the factors that help in binding of these transcription factors, general transcription factors, to initiate the transcription at the promoter region? These are the general transcription factors, and these are their functions and how many number of subunits are there. These are all required for initiation of transcription, and TF2H is the most important function. It unwinds the DNA and phosphorylates, but others also, of course, have important functions to do. And for the elongation, there are other proteins which help in the elongation of the RNA polymerase II on the template. So there are many other transcription factors apart from the general transcription factors that are required to initiate transcription of any gene in case of eukaryotes. This is one example of PEP carboxykinase promoter region. So it's showing the complexity of regulatory input to this gene. So many transcription factors you can see over here, they are needed. They are required for this. So many number of transcription factors are there. So these are all, and these are they allow the binding of this general transcription factor. So in this lecture I will discuss how this interaction takes place. So basically the general transcription factors, they no doubt initiate the transcription, but before that there are some specific transcription factors and certain other proteins that are required. This I already have discussed, that these how this chromatin structural changes that help in the initiation of transcription. The successful binding of active RNA polymerase II holoenzyme to the promoter requires action of other proteins of three types. As I mentioned, these are the general or basal transcription factors, TF2A, 2F, 2H, and etc. And then there are DNA binding transactivators, which bind to enhancers or upstream activator sequences and facilitate the transcription. So these DNA binding transactivators basically bind to certain region on the DNA which help in the transcription, initiation of transcription. And then coactivator. This acts indirectly, not by binding to the DNA, but it is required for communication between DNA binding transactivators, number 2 above, and the RNA polymerase II general transcription factors, number 1 above. So for 1 and 2 to interact, 1 and 2, coactivator is required, which mediates this interaction between basal transcription factors and DNA binding transactivators. So here is the overall picture. This is the Tata box at the promoter region, and this RNA polymerase along with its transcription factors, it will bind here, and this is the CTD of RNA polymerase II, which is phosphorylated. But before that, there are certain other proteins such as HMG proteins. These are required. Then modification and remodeling enzymes are required. All these help in the opening of the chromatin structure for transcription machinery to bind to. And then there are enhancer elements, which are DNA sequences at various places. To these enhancer elements or upstream activation sequences, these are also called as upstream activation sequences. To these bind these transcription activators. So this one, pinkish type, is protein binding domain, and this one, the greenish one, is the DNA binding domain of these transcription activator proteins. And these transcription activator proteins tells when to start the transcription. And this transcription initiation is mediated by this mediator, which these are certain proteins which act as communication between transcription activators and the general transcription factors. So transcription activators communicate with the mediator proteins that ultimately ask the RNA polymerase factors to bind and initiate transcription. And in case of repressors, there may be, although there are not much repressor which exist in case of eukaryotes, but if there are some, they inactivate the activators and also they interact with the mediator, or they may interact with the general transcription factor to stop this transcription. So overall we can see the sequence of events. Activator bind to the enhancer elements, then remodeling and mediators, and then general transcription factors, and finally initiation of transcription. So here activators, this picture is more clear. You can see first activator binds to the enhancer elements, and then mediator comes and modification and remodeling enzyme come. So they come at this place, and finally this general transcription factor come, join here and start the transcription. The general transcription factors, these are the same example that multiple transcription factors are there which initiate this transcription to occur. So in this particular lecture I will give a couple of examples of this activation at this place, but there are certain other signal molecules which give the signal when this activator is going to come on this UAS, or upstream activating sequence. So that I will discuss in my part 2 lecture of this eukaryotic gene expression. Here is one example in case of eukaryotes from yeast from the galactose metabolism. So for galactose metabolism, this is the US upstream activator sequence, which can be bound by gal4p, which is an activator. Gal4p is the activator, and this gal ATP is the inhibitor of this gal4p. So gal3p plus galactose along with gal3p, they induce this system, and this gal3p takes out this gal ATP and export it to the cytosol. So as soon as this is released, this gal ATP is released, the other proteins like mediator and SAGA, these are set of proteins which are required for this modification and remodeling. They bind at this place, and mediator comes and transcription is initiated. So this galactose metabolism in yeast, where multiple genes, these are the regulated genes which are controlled by these regulatory genes gal3, gal4 and gal80, by these are inducer, activator and inhibitor respectively. So this here it is being shown that what are specifically these proteins are, like GC, and this already I have discussed these proteins in the chart in the table earlier, and what is the sequence by which they bind, like 2A, then theta binding, then mediator, and what other proteins like SW1, SNF and SAGA proteins and how they are assembled at this place for the galactose metabolism, and finally this transcription is initiated. Uh, as far as different UAS are concerned, the famous or the most common ones are the CCAT is one of these, and this CCAT is the sequence on the DNA. US, this already I have shown the galactose metabolism, and then there is a GC. So all these sequences have specific activators which bind to, like this is glutamine rich shown by Q, and this is acidic domain, its active domain is acidic domain, and this is the proline rich in the CTF1. So what they do is they activate specific genes. Now it depends if SP1 binds to this GC, it has glutamine rich domain and it can activate certain types of genes. Similarly, CTF1, it binds to CCAT and it can its proline rich domain activate certain genes, and similarly this UAS and GAL4P, as I mentioned this example earlier. And we can also swap this, like in this case SP1 binds to the GC, but this is SP1. All these activators have two domains. One is the DNA binding domain, and other is the protein binding domain. So if we change the protein binding domain of SP1 with the CTF1, then it will activate the genes relevant to the CTF1, not with the SP1. So genes which are, because it will allow for certain mediator proteins which according to this one. So this is more clear picture of these CCAT, US and GC, and these are the activators as I mentioned previously. So this basically is called as domain swapping. So this domain can be swapped with some other protein. Another example, very common example, is replacing DNA binding domain of GAL4P with DNA binding domain of E. coli lactase repressor. So if we replace the DNA binding domain of GAL4P with DNA binding domain of E. coli lactase repressor, we won't see any binding at US and neither activation of the GAL genes because of this, because it won't bind there, because this contains the DNA binding domain of lactase repressor. But activation of genes may take place when UAS sequence is replaced with LAX-A recognition sequence in the DNA binding domain. So in this way, you can devise some interesting experiments to check these interactions by domain swapping. So I end the first part of eukaryotic gene expression here. We saw that what were the differences between eukaryotic and prokaryotic gene expression, and how this eukaryotic gene expression goes on with the general transcription factor, but it is backed by the specific transcription factors and different enzymes and proteins which are related to histone and chromatin remodeling and modification, and then mediator is required for the communication, and DNA binding elements, and they're attached to the activators having two domains, DNA binding domain and protein binding domain. Protein binding domain attaches to the or have a communication with the mediator, and domain swapping are quite interesting experiments where we can change different types of DNA binding and protein binding domains to see what results are. In the second part of this, I will discuss what are the certain proteins which are required for activation in the that is the specific transcription factor, or what specific proteins are there in the transcription activation. And also what are certain factors or signals which are required for initiation of transcription beyond this activator. What are those signals which give signal to these activators to bind to the upstream activator sequences or elements, DNA elements, to initiate the process.